In an earlier lesson, we saw that cells need a large surface area to their membrane in order to increase the surface area for material exchange. But it's also helpful for cells to have a large surface area inside. And so in order to do this, many cells have structures called organelles. And these organelles will increase the efficiency of things that happen inside the cells. In this video, we're going to take a look at the various organelles, what they do, and how they help the cell maintain homeostasis. Let's do a quick review of cell types. Remember, there are two main categories, prokaryotic cells, like bacteria, and eukaryotic cells. And for these two types, ribosomes can be found in all of the cells. However, for eukaryotic cells, that's where we're going to find the membrane-bound organelles that really help increase efficiency of the cell. And eukaryotic cells can include animals and plants. So for this video, we're going to be focusing on eukaryotes. Now, why are organelles such an advantage? Well, if you think about a business, and you go inside a business, you'd probably see the office organized into cubicles with lots of different desks and areas for different people with different jobs to get their work done. Well, it's the same thing with a cell that's divided up into smaller components called organelles. Those different organelles, first of all, are specialized for different functions, but they also increase the surface area for chemical reactions to occur. And they isolate chemical reactions. Just like people doing different jobs sometimes benefit from having their own space to do their work. So all of these things improve efficiency. So let's take a look at the various organelles we would find in a eukaryotic cell. The first group of organelles are all related to production, primarily production of proteins, but some other things as well. So this all begins in the nucleus, which we're pretty familiar with. The nucleus has the hereditary information, or DNA, inside of it. And it's also surrounded by a double membrane known as the envelope. There are pores in the envelope, here, all over, to allow RNA to exit the nucleus. The pores also allow other things in and out. And then within the nucleus, we have the nucleolus. And the nucleolus makes ribosomes. So speaking of ribosomes, we're pretty familiar with them. They're very simple, small organelles. And they are where protein synthesis happens. They're also made of two subunits that will interact together at the start of translation. And free ribosomes, the ones that are floating around in the cell, will produce proteins that stay in the cell. That's an important point. Now, the rough endoplasmic reticulum is attached to the nucleus and other organelles. And here we can see the rough endoplasmic reticulum is studded with ribosomes. And those ribosomes that are stuck in the rough endoplasmic reticulum make secretory proteins. Secretory proteins are proteins that are going to be released from the cell. For example, a hormone like insulin. And the ER also creates smaller compartments within the cell, just like cubicles help people get their jobs done. The ER will also help transport materials through the maze-like fold of membranes. The smooth endoplasmic reticulum, which is continuous with the rough endoplasmic reticulum, is smooth because it lacks ribosomes. And the main job of the smooth endoplasmic reticulum is to make lipids. However, it also will break down certain toxins like alcohol. Alcoholics often have large amounts of smooth endoplasmic reticulum in their liver cells, attempting to break down all the alcohol that's consumed. The Golgi apparatus is another series of membranes that's not connected to the ER, but is often found close to the ER. And you can see from the structure that it's basically a stack of sacks, kind of looks like pancakes. And what they do is they package and they process materials. For example, they might add little protein or carbohydrate tails onto proteins to make a glycoprotein. And once the proteins or the polymers are all finished, a vesicle will carry the product off somewhere else in the cell to be used. The Golgi apparatus also makes lysosomes. Here you can see a lysosome budding off, and we'll talk more about that in a second. So let's sum all of that up by tracing the path of a secretory protein, starting with the nucleus. In the nucleus is where the DNA would be translated into mRNA. Once we've got the mRNA will leave through the nuclear pore, and then it will head to a ribosome in the rough ER. And that ribosome is going to translate the mRNA into protein. From there, the protein is going to be transported in a vesicle to the Golgi apparatus. Then the Golgi apparatus is going to put the finishing touches on the protein. Once the protein is all finished, another transport vesicle will carry it to the cell membrane. And there, via exocytosis, the cell membrane can secrete or release the protein. And again, that protein could be a hormone like insulin. Now let's take a look at energy organelles, because all of these processes require energy. Well, the chloroplast is an organelle that we would find in any autotrophic eukaryotic cell, so a plant cell or a protist cell like an algae. And the job of the chloroplast is to convert solar energy, sunlight, into the chemical energy of food, namely glucose. And it does this by also taking in CO2 and water, and it releases oxygen as a waste. Now, if you look at the structure of the chloroplast, the structure helps it do this job well. First of all, it's got ribosomes, so that it can make enzymes to do these reactions. It's also got its own DNA. And then within the chloroplast, we see stacks of green pancakes called thylakoid, and each stack is a granum. And those increase the surface area for the photosynthetic reactions to occur. And then finally, the green color is due to chlorophyll, a green pigment, which we'd find in here, and that pigment's job is to actually capture the sunlight. The mitochondrion is also an energy-producing organelle, but unlike the chloroplast, we would find this in all eukaryotic cells, plants, animals, fungal cells, protist cells. And the job of this is to transform the chemical energy in food, namely glucose, into usable energy, ATP. And during that process, it also takes in oxygen, and it releases water and CO2 as a waste. Just like the chloroplast, we can see that there's some special structures in the mitochondria. There's ribosomes to make enzymes, there's circular DNA, and there's also folds within the mitochondria. The inner membrane folds into these cristae, and what these do is they increase the surface area for chemical reactions to occur, just like the cubicles or desks in an office. And the cristae have enzymes embedded in them in order to perform cellular respiration. Lastly, let's look at the custodial organelles. These are organelles responsible for storage, for cleanup, for keeping things tidy. The key one is the lysosome, which we've seen before. The lysosome is basically a small sac full of enzymes. Those enzymes are hydrolytic, meaning they digest things. And the things they digest could be waste, pathogens, or old cell parts. And this is important because it helps the cell recycle materials. It could recycle old organelles, or it might recycle entire cells during the process of apoptosis, which we learned about last unit. And here is that process in action. Here we've got some food, or could be a pathogen being taken in by the cell. It's stored briefly in a vacuole. A lysosome comes along and fuses with it, and the enzymes in the lysosome are going to break that material down. Similarly, here we've got a vesicle which contains a damaged mitochondrion. Now, in order to reuse it, the lysosome again will fuse with the vesicle, and it'll break down the mitochondria into smaller parts that can then be reused. Last but not least, the central vacuole, which we find in plant cells. Its job is to store food, water, waste, all sorts of things. And it also has enzymes that can break things down. The other important thing about the central vacuole is that when it's turgid, when it's full of water, it will support the plant cell. And also keep in mind that animal cells have small vacuoles for storage as well. And that concludes our whirlwind tour of the organelles that you would find in eukaryotic cells.